Amino acids in the regulation of aging and aging-related diseases. Abstract. Amino acids are the building blocks of protein, but also play important cellular signaling roles. The mechanisms through which altered levels of many amino acids are sensed and the signals transmitted are still largely unknown. Increasing evidence is showing that these signals may influence the aging process. In this regard, methionine restriction appears to be an evolutionary conserved mechanism to delay aging, and supplementation with glycine can mimic methionine restriction to extend lifespan in rodents. Tryptophan restriction may also activate specific anti-aging pathways, but it could interfere with cognitive function. With exercise, the consumption of dietary branched-chain amino acids, BCAAs, may be beneficial in building muscle mass, but high levels of BCAAs as well as tyrosine and phenylalanine in the bloodstream are associated with metabolic disease such as insulin resistance. Individual supplementation or restriction of several different amino acids has shown promise in the treatment of insulin resistance in rodents. Much progress regarding the effects of amino acids on longevity has been made using yeast, nematodes, and fruit flies. Clearly, much more research is needed to understand the signaling pathways activated by amino acid imbalance before efficacious and well-tolerated dietary interventions can be developed for human aging and aging-related disorders. In this review, the mechanisms through which altered dietary and cellular levels of the 20 proteogenic amino acids affect aging or aging-related disorders are discussed. 1. Introduction. There are 20 proteogenic amino acids used as the building blocks of protein synthesis. Plasma amino acid imbalance can result from liver or kidney disease and may lead to some of the phenotypes of those diseases. 1. Roughly 40 years ago, amino acid solutions were formulated to treat such imbalances. 1. However, an imbalance of amino acids can also lead to the activation of stress response pathways and lifespan extension in model organisms. 2, 3, 4. Dietary intake of large amounts of a single amino acid can be toxic. 5, 6. Single amino acids have been administered to humans without severe side effects in the 3 to 40 g per dose range, with cysteine and methionine being the most toxic. 1. However, rodents showed a slowed rate of growth when large amounts of a single amino acid were given with a low-protein diet. 1. Rodents can sense the lack of a single amino acid in their food using the anterior piriform cortex region of the brain and will become anorexic on such a diet. 1. There are mechanisms present to detect unchanged ARG-tRNAs and activate the eukaryotic initiation factor 2-alpha, EIF2-alpha, kinase general amino acid control non-depressible 2, GCN2, to signal the decreased level of a specific amino acid so the cell can decrease the rate of protein synthesis accordingly. 7. The other major pathway for sensing the level of single amino acids and adjusting the rate of protein synthesis is the target of rapamycin, TOR, pathway 8. Modulation of either of these pathways in C. elegans can extend lifespan in part due to increases in proteostasis. 9. As decreased protein synthesis was found to correlate with increased lifespan under several experimental conditions. 10, 11. But other mechanisms such as activation of the Fa4/FOXA transcriptional regulator 12 and the prevention of aging-induced mitochondrial fission 13 to maintain energy levels 14 also likely play an important role in lifespan regulation by TOR and GCN2. Since rapamycin, a TOR inhibitor, can extend the lifespan of mice 15, these pathways are also important targets for the regulation of mammalian longevity. When amino acid intake exceeds the requirements for protein synthesis, they are deamined and catabolized into pyruvate, acetyl-CoA, or mitochondrial citric acid cycle intermediates for energy generation. 16. In the nematode C. elegans, individual supplementation at an optimal concentration with the vast majority of amino acids or citric acid cycle intermediates extended lifespan. 3, 17. Higher concentrations frequently decreased lifespan, suggesting a hormetic dose response. Supplementation with peptone, a balanced source of amino acids, decreased lifespan when C. elegans was cultured in liquid media. 3, 18, but not when cultured on agar plates. 19. suggesting that the effects of high amino acid levels depend upon the culture conditions. Like mammals, C. elegans evolved to utilize several D-amino acids as signaling molecules and can metabolize a subset of these D-amino acids for energy generation. 20,21. Individual supplementation to C. elegans with each of the three D-amino acids, D-alanine, D-aspartate, or D-glutamate, which are metabolized by the worms and present in the E. coli that the C. elegans standardly consume as the major portion of their diet. 21. Also extended lifespan. 3, while supplementation with D-proline or D-serine, which are not readily metabolized by the four characterized C. elegans D-amino acid oxidizing enzymes. 20,21, did not extend lifespan. 3. This data suggests metabolism as an important component of the longevity effects of supplemented amino acids. However, other data suggest that metabolism is not always required for amino acid-mediated longevity in C. elegans, as increasing the intracellular levels of tryptophan 22 or branched-chain amino acids, BCAAs, isoleucine, leucine, and valine, 23 by preventing their catabolism increased lifespan. So, clearly different signaling pathways are activated by different amino acids in the regulation of lifespan. Whether or not individual amino acid supplementation at an optimal dose can influence the lifespan of other multicellular organisms such as fruit flies, rodents, or humans is mostly unknown. But recently it was shown that glycine supplementation increased the lifespan of mice. 24, and the molecular mechanism is likely similar to that induced by methionine restriction. 25. Calorie restriction, CR, or dietary restriction, DR, is an evolutionary conserved mechanism to delay aging resulting in lifespan extension. In fruit flies, the lifespan extending effects of DR have been shown to be regulated by the protein to carbohydrate ratio. Adding back essential amino acids to dietary restricted flies prevented lifespan extension. 4. This area has been extensively reviewed. 26, and will not be covered here in detail. Decreased levels of single amino acids in the culture media also extended replicative. 27, or chronological, Table 1, lifespan in yeast. Replicative aging in yeast appears to be due to a loss of vacuole acidification that leads to the vacuolar release of neutral amino acids that reduce mitochondrial membrane potential. This aging-induced loss of vacuolar acidification is delayed by CR28. One large study using a geometric framework of nutrition approach in mice also implicated decreased protein levels as having an important role in the longevity effects of CR29. But these results could be due to the unique method, using cellulose to fill the void of restricted carbohydrate, lipid, or protein food mass, of implementing the restricted diets, as many older studies using rodents have shown that protein restriction is not responsible for the large, 30%, increase in median lifespan of the typical 30%, 40% CR diet 30. But protein restriction of 50%, 85% can also increase lifespan up to roughly 15%, apparently independently of the effects of CR. Many beneficial disease-delaying effects of protein restriction have been found. 31. Since amino acids signal through distinct pathways to modulate lifespan in model organisms, consuming an optimized ratio of amino acids in the diet may be a viable approach for delaying aging in humans as well. Along these lines, research using fruit flies and mice has shown that consuming a diet where the amino acid content matches the in silico translated codon usage of the expressed genome, the exome, enhances the rate of growth and development without affecting lifespan. 32. Therefore, the levels of amino acids in the diet may also one day be optimized for other purposes, including increased stress resistance or a decreased rate of aging. This could be particularly useful for the treatment of diabetes, where the plasma levels of all three BCAAs and phenylalanine and tyrosine positively associate with the disease. 33. This may somehow be related to the finding that the level of these same 5 amino acids plus glutamate increased in brain when rats were fed diets of altered protein content, while the levels of other amino acids changed little to none. 34. More research is needed on the ability of amino acid supplementation to alter levels of free amino acids in mammalian brain regions. When examining the fibroblasts taken from 15 primate species, 33 bird species, and 13 rodent species, free levels of 11 of the 20 amino acids including arginine, glutamate, histidine, leucine, lysine, methionine, phenylalanine, proline, tryptophan, tyrosine, and valine were found to have a positive association with species lifespan 35. Individual supplementation with 18 of the 20 amino acids extended lifespan in C. elegans, Table 1. These data suggest that free amino acids have an important role in the regulation of aging. Interestingly, 8 of the 20 amino acids, including 5 of the 11 listed above, were able to protect Caco2 intestinal epithelial cells from oxidative stress including alanine, cysteine, histidine, isoleucine, leucine, lysine, tryptophan, and valine 36. 5 of the amino acids were the abundance associates with longevity that did not show an antioxidant effect in Caco2 cells including arginine 37, glutamate 38, methionine 39,40, proline 41,42, and tyrosine 43,44 have shown antioxidant effects using in vitro assays or have been shown to stimulate the expression of antioxidant enzymes in other cell types or tissues. These data suggest that decreased oxidative stress may play a role in the positive association between free amino acid levels and longevity. In the sections below we outline the current state of knowledge regarding how altered levels of each of the 20 proteogenic amino acids affect aging, aging-related diseases, and the associated signaling pathways. 2 amino acids. 2.1. Alanine. Alanine is a non-essential amino acid and also one of 13 exclusively glucogenic amino acids 45. Following its uptake and entry into the hepatic circulation, alanine is taken up by the liver, where much of the alanine pool is converted to pyruvate, which enters into gluconeogenesis. In a fasted state, skeletal muscle releases large amounts of alanine and glutamine into the circulation where they are taken up by the liver and kidney and used for gluconeogenesis 46. Increased alanine consumption may be beneficial for metabolic disorders such as type 2 diabetes as its supplementation to a cultured pancreatic beta cell line increased both non-oxidative and oxidative glucose metabolism and the secretion of insulin 47. Consistent with this, alanine infusion into hyperglycemic dogs increased insulin secretion without much change in glucagon levels, but in the fasting state when glucose levels were low, alanine infusion stimulated glucagon secretion, while having little effect on insulin levels 48. In mice, dietary alanine was shown to prevent obesity due to a high-fat diet 49. Alanine supplementation has also been shown to stimulate the proliferation of lymphocytes 50 and thymocytes 51, which may decrease the aging-related loss of immune system function. A diet containing 10% alanine administered to rats was also able to prevent the formation or dissolve pre-existing kidney stones 52. Alanine levels have been shown to decline with aging in mouse plasma 53 and muscle 54. Alanine, leucine, and glutamine levels were shown to decline in aged parasitoid wasps 55. In yeast, deletion of the ALT1 alanine transaminase gene increased alanine levels and decreased chronological lifespan, while longevity extending CR decreased alanine levels 56. In C. elegans supplementation with 1 mm or 10 mm alanine extended lifespan 3, while supplementation with 5 mm did not 3,23, Table 1. The metabolism of alanine to pyruvate may play a role in the extended longevity of nematodes as pyruvate supplementation also extended lifespan 57. 2.2. Arginine. Arginine is a semi-essential amino acid and the anti-aging effects of arginine supplementation have been reviewed 58. Arginine catabolism is altered in learning and memory centers in the aged brain 59. Arginine has been shown to stimulate the proliferation of young human T lymphocytes to aid in wound healing 60,61, but it did not stimulate the proliferation of young or aged rat lymphocytes 62. Increased arginine levels resulted in a switch from glycolysis to oxidative phosphorylation in activated human T lymphocytes and an upregulated anti-tumor response directly associated with their enhanced survival. 63. Arginine was shown to reverse the aging-related loss of acetylcholine-induced vasodilation of coronary endothelial cells in humans. 64 and to improve depressed macrophage and wound immune function following hemorrhage in rats. 65,66. Long-term arginine supplementation prevented the aging-related decrease in renal function. 67. The protective effects of arginine may be due in part to its metabolism to polyamines that are known to have anti-aging effects. 68. Consistent with this mechanism, anti-aging DR has been shown to increase the levels of polyamine synthesis enzymes through a post-transcriptional mechanism. 69. Arginine is used for nitric oxide synthesis. 70 and decreased nitric oxide synthesis contributes to the aging of the cardiovascular system. 71. Arginine supplementation has been shown to increase the secretion of insulin-like growth factor 1, IGF-1, 72 and growth hormone. 73, both of which have been shown to have pro-aging functions in mice. 74,75. Arginine is hydrolyzed by the enzyme arginase to form ornithine and urea in the liver during urea cycle function. 76 and in the liver and other tissues for polyamine synthesis from ornithine. The role of arginase in aging has been reviewed. 77. Arginine stimulates mechanistic target of rapamycin, mTORC1, kinase activity by two different mechanisms. 78,79. First, cytoplasmic arginine binds the castor-1, cytosolic arginine sensor for mTORC1 subunit 1, protein to stimulate mTORC1. Second, the SLC38A9 lysosomal amino acid transporter, that transports leucine, phenylalanine, isoleucine, tryptophan, methionine, valine, and tyrosine 80, interacts with the regulator protein complex that signals to mTORC1 dependent upon high, likely lysosomal, arginine levels. 81. The activation of mTORC1 leads to increased protein synthesis and inhibition of autophagy that may exert a pro-aging effect in many cellular contexts. Increased arginine levels were found in aged Drosophila. 82. In yeast, deletion of the arginine permease can one extended replicative lifespan depending upon the transcriptional regulator GCN4 and translational activation of stress response genes. 83. Arginine supplementation potently increased the lifespan of C. elegans, Table 1, 3. Although C. elegans lacks a castor-1 homologue, its genome does contain homologues to the lysosomal amino acid transporter SLC38A9 and the regulator complex protein LAMDR2 that may play a role in signaling cellular arginine levels. 2.3. Asparagine. Asparagine is a non-essential amino acid except when glutamine levels are very low. 84. Asparagine synthetase catalyzes the cytoplasmic synthesis of asparagine from aspartate and ammonia. Older subjects showing physical frailty and sarcopenia were shown to possess increased plasma levels of asparagine, aspartate, and glutamate. 85. Asparagine plays a role in the proliferation of cancer cells via activation of mTOR during tumor development. 86. Consistent with this role, a diet low in asparagine has been shown to be protective against breast cancer metastasis. 87. Asparagine and glutamine metabolism play essential roles in blood vessel formation. 88 and tumor growth. 89. Asparagine and glutamine also play anti-inflammatory roles to protect intestinal integrity and stimulate intestinal epithelial cell division to protect against lipopolysaccharide, LPS, treatment. 90. Treatment with asparagine and aspartate together decreased fatigue and increased time to exhaustion during moderate or intense exercise. 91,92. In yeast, removal of asparagine from the media extended the chronological lifespan by inhibiting TOR kinase. 93. The asparagine catabolizing enzyme asparaginase was shown to be selectively retained in yeast mother cells following the budding of daughter cells. 94. Largely explaining the decrease in asparagine levels in replicatively aged yeast. 95. Asparagine supplementation only slightly extended mean lifespan in C. Elegans at a 5 mm dose and decreased lifespan to the greatest extent of any amino acid at a 10 mm dose, Table 1, 3. 2.4. Aspartate. The non-essential amino acid, aspartate, participates in a range of important cellular functions such as the urea cycle and the malate aspartate shuttle, which transports nicotinamide adenine dinucleotide, NADH, reducing equivalence between the cytoplasm and mitochondrial matrix. The shuttle plays an essential role in CR-mediated longevity in yeast by increasing the nucleocytoplasmic NAD+/NADH ratio to activate sirtuin deacetylase 96. Aspartate is synthesized from oxalostate by aspartate aminotransferase. Mitochondrial electron transport chain function declines with aging 97 and is required for aspartate and pyrimidine synthesis 98. Metals such as Mn2+, Cu2+, Zn2+, and Mg2+ bind to aspartate to form complexes with antioxidant function 99. Aspartate, as well as proline and serine, supplementation was effective at decreasing reactive oxygen species, ROS, production and increasing ATP levels in M17 neuroblastoma cells expressing alpha-synuclein treated with ferrous sulfate 100. Aspartate or glutamate was effective in promoting blood antioxidant levels when male pigs 101 or weaned piglets 38 were challenged with hydrogen peroxide. However, glutamate, but not aspartate, was effective at reducing diquat-mediated oxidative stress in piglets 102. Aspartate supplementation also decreased ethanol-induced hepatotoxicity and oxidative stress in the testes of rats 103. Within the brain, aspartate acts as an agonist of N-methyldiaspartate, NMDA, receptors to regulate neurotransmission 104. Therefore, increased amounts of aspartate may contribute to aging-related cognitive impairment by facilitating excitotoxicity 105. Restriction of aspartate was shown to increase the chronological lifespan of yeast 106, likely by inhibiting TOR kinase activity 107. However, the replicative lifespan of many yeast gene deletion strains positively correlated with increased levels of aspartate, methionine, proline, threonine, isoleucine, histidine, and glutamine 108. So some amino acids, such as aspartate, may show opposite effects on the chronological and replicative lifespan of yeast. Aspartate was one of two amino acids that did not extend lifespan when supplemented in the 1 to 10 mm range to see Elegans, Table 1, 3. 2.5. Cysteine. Cysteine is a non-essential amino acid. Total cysteine, including reduced cysteine and oxidized cysteine, and the cysteine to cysteine ratio in human plasma were shown to increase with aging 109, while the levels of some amino acids were shown to decline 110. The plasma cysteine/cysteine oxidation state is an important determinant of systemic inflammation 111, which likely contributes to aging 112. A reduced extracellular cysteine/cysteine oxidation state was also shown to restore free mitochondrial NADH levels in isolated neurons from aged mice and the 3xTGAD Alzheimer's disease mouse model 113. Mitochondrial cysteine oxidation may play a role in the aging process as long-lived animals have evolved to encode less cysteines in the genomes of their mitochondria, where ROS production is the highest 114. Cysteine supplementation has shown promise as an adjuvant for type 2 diabetes 115. When rats were fed a low-protein diet, cysteine supplementation was shown to decrease the plasma levels of homocysteine 116, a risk factor for neurodegenerative and cardiovascular diseases 117. However, high levels of plasma cysteine have been associated with many disorders including coronary heart disease, Alzheimer's and Parkinson's diseases 118, rheumatoid arthritis 119, and systemic lupus erythematosus 120. Systems biology experiments in yeast identified that high cysteine levels inhibit protein synthesis to cause toxicity and that supplementation with leucine and pyruvate prevented this toxicity. The protective effect of leucine relied upon its transamination to alpha-ketoazocaprote, KIC, which depended upon the function of the tRNA methyltransferase NCL1-121. The antioxidant glutathione is composed of cysteine, glutamate, and glycine 122. The oxidized glutathione disulfide, GSSG, to reduced glutathione, GSH, ratio increases with aging, but is not in strict equilibrium with the cysteine/cysteine ratio 123. When GSH is consumed orally by humans, it is hydrolyzed in the intestine by gamma-glutamyl transferase, and plasma GSH was not altered 124. Supplementing with cysteine and glycine together has shown more promise and led to enhanced plasma GSH levels in aged subjects 125. This elevation corresponded with a marked decrease in oxidative damage. N-acetyl-L-cysteine, NAC, is a membrane-permeable prodrug form of cysteine that releases cysteine through the action of intracellular esterases. NAC supplementation has been shown to extend the lifespan of mice 126, fruit flies 127, and nematodes 128,129. But the effects were gender-specific and not concentration-dependent, suggesting hormesis 127. The negative effects at higher doses may also suggest reductive stress 130. Studies using NAC have been reviewed 131. Consistent with the effects of NAC, cysteine supplementation potently increased lifespan in C. elegans at 2 lower doses, but not at the highest 10 mm dose administered, Table 1, 3. 2.6. Glutamate. Glutamate is a non-essential amino acid most well known for its role as an excitatory neurotransmitter in the brain. Therefore, glutamate plays an essential role in learning and cognition, but at high levels glutamate induces neuronal excitotoxicity, contributing to neuronal injury in neurodegenerative disorders including Alzheimer's disease, AD, 132, and amyotrophic lateral sclerosis, ALS, 133. Glutamate levels have been shown to decline in human brain during young adulthood 134. Glutamate is only very slightly permeable through the blood-brain barrier, BBB, so relatively high levels of dietary glutamate are non-toxic in healthy subjects. But it is not recommended to consume membrane-permeable glutamate esters that may permeate the BBB. Patients with neurodegenerative disorders or who have had a stroke may also choose to limit their glutamate consumption as their BBB may be more permeable 135, and lower plasma glutamate levels may stimulate limited release of glutamate from the brain to decrease excitotoxicity 136,137. The crucial balance in the brain between low extracellular glutamate concentrations and high intracellular glutamate concentrations is kept in check by plasma membrane glutamate transporters. Astrocytes take up glutamate, convert it to glutamine, and release the glutamine to the extracellular space where it can be taken up by neurons that convert it back to glutamate for release at synaptic terminals 138. One potential therapy for lowering systemic glutamate levels in neurodegenerative disorders and stroke is the infusion of oxalostate and the aspartate transaminase enzyme 136,137. However, glutamate plays important roles in the body, such as binding free ammonia, which is especially toxic to neurons 139, and it serves as an important anaplerotic source of the anti-aging citric acid cycle intermediate alpha-ketoglutarate 140. Higher levels of glutamate were found in the plasma of long-lived naked mole rats compared to mice, while lower levels of many other amino acids including glycine, methionine, leucine, tyrosine, phenylalanine, and tryptophan were found 141. Another group found glutamate, alanine, aspartate, isoleucine, leucine, serine, and threonine levels to be higher in the plasma of naked mole rats than mice 142. One study found that high glutamate levels increased the chronological lifespan of yeast 106. However, another group using different experimental conditions found removal of glutamate from the media to extend chronological lifespan 93. In C. Elegant studies, glutamate was one of the most potent lifespan-extending amino acids when supplemented at the 1 and 5 mm doses, but it decreased lifespan when supplemented at the 10 mm dose, Table 1, 3. Knockout of the glutamate transporter GLT4 decreased nematode lifespan, while knockout of the glutamate transporter GLT5 increased lifespan 143. 2.7. Glutamine. Glutamine is an essential amino acid and the most abundant amino acid in the serum, CSF, and muscle. Its anti-aging roles have been reviewed 144. Glutamine is an important energy source, especially in rapidly dividing cells such as epithelial cells and immune cells, due to its ability to be readily broken down and feed carbon into alpha-ketoglutarate in the citric acid cycle 145. Increased glutamine levels can inhibit fatty acid oxidation, decrease plasma glucose, and attenuate weight gain in mice prone to obesity 146. A mitochondrial glutamine transport activity has been characterized, but its molecular identity is still unknown 147. Following prolonged exercise by athletes supplemented with glutamine, an increased T helper/T suppressor cell ratio was measured, which corresponded with a decrease in the rate of infection 148. Glutamine regulates the expression of genes involved with cellular proliferation and survival, as well as decreases the expression of pro-inflammatory genes such as NF-beta 149. Supplementation with glutamine increased the levels of anti-inflammatory monocytes and regulatory T lymphocytes in the serum of diabetic mice, contributing to enhanced muscle regeneration following limb ischemia 150. Alanine facilitates glutamine metabolism. The combination of glutamine and alanine was shown to reduce the glutathione couple, GSSG/GSH, 151 to increase antioxidant function and had anti-inflammatory and cytoprotective effects in rats undergoing resistance exercise 152. Long-term supplementation with glutamine was shown to be beneficial for maintaining intestinal immunity in the elderly 153. Glutamine synthetase is highly expressed in the mammalian brain 154, where it may play a neuroprotective role in healthy young animals 155. Glutamine levels and glutamine synthetase activity are decreased in Alzheimer's disease brain, but glutamine synthetase was increased in a fraction of hippocampal neurons where it can play a maladaptive, cytotoxic role when glutamine levels are low 156. Consistent with this, glutamine supplementation was shown to protect against Alzheimer's amyloid beta toxicity in cultured neurons 157. The role of amino acids in Alzheimer's disease brain has been reviewed 158,159. In yeast, inhibition of glutamine synthesis decreased glutamine levels 160 and increased chronological longevity 93. During chronological aging, the intracellular levels of 16 of the 20 amino acids were found to decrease, with glutamine levels declining to the greatest extent, followed by glutamate, lysine, histine, methionine, and cysteine 161. Supplementation of glutamine to C. Elegans had an almost identical effect on lifespan as glutamate at all three doses administered, Table 1, suggesting that the glutamine synthetase and glutaminase enzymes readily interconvert these amino acids when they are consumed in the diet. 3. 2.8. Glycine. The amino acid glycine is considered conditionally essential as it can be synthesized from serine, but not always at the levels needed. The known effects of dietary glycine supplementation have been reviewed 162. As mentioned earlier, glycine is the one amino acid studied thus far in which supplementation has been shown to extend the lifespan of mice, albeit moderately. 24. Dietary glycine supplementation also extended the lifespan of Fisher 344 rats through a mechanism mimicking methionine restriction to increase the hepatic clearance of methionine 163. Glycine supplementation has been shown to restore T cell activation, T cell 1-carbon metabolism, and mitochondrial function in aged mice 164. Administration of glycine was also shown to decrease oxidative stress and inflammation by decreasing the levels of advanced glycation end products, AGEs, by inducing the expression of glyoxylase 165. By comparing young and old human fibroblasts, it was found that epigenetic downregulation of the glycine C-acetyltransferase, GCAT, and serine hydroxymethyltransferase 2, SHMT2, genes involved in mitochondrial glycine synthesis, see Fig 1, are involved in the aging-related loss of cellular respiration. Adding glycine to the culture media restored the phenotype of the aged cells back to that of the young cells 166. Supplemental glycine can be transported across the mitochondrial inner membrane by the mitochondrial carrier family member SLC25A38-167. Another study did not find much change in human whole-body glycine metabolism with aging, but found large changes following altered protein consumption 168. Glycine is an inhibitory neurotransmitter that plays especially important roles in the spinal cord, brainstem, and retina 169. Increased glycine levels can induce hypothermia and sleep by binding NMDA receptors in the suprachiasmatic nucleus of the brain 170. Glycine supplementation protected the colon wall during radiotherapy 171. Glycine infusion led to vasodilation in the rat kidney, but this effect was greatly reduced in aged animals 172. N-arachidone oil glycine and other N-acyl amino acids play important anti-inflammatory and analgesic functions 173,174. Therefore, therapies using these compounds could be tested for improving inflammation in aging-related disorders. 1. Fig. 1. Serine, glycine, threonine, methionine, cysteine, and histidine metabolism influence 1-carbon and NAD-redox metabolism. TDH and GLY1 enzymes are not present in humans. Metabolites shown on the mitochondrial, inner, membrane can be transported into and out of the matrix space. Glycine catabolism by the mitochondrial glycine cleavage system is not shown. This results in the conversion of THF to 5,10-MeTHF for additional 1-carbon metabolism and is accompanied by the reduction of NAD+ to NADH. THF, tetrahydrofolate, 5-MeTHF, 5-methyl-tetrahydrofolate, 5,10-MeTHF, 5,10-methylene tetrahydrofolate, CoA, coenzyme A, NAD+, nicotinamide adenine dinucleotide, oxidized, NADP+, nicotinamide adenine dinucleotide phosphate, oxidized, NADH, nicotinamide adenine dinucleotide, reduced, NADF, nicotinamide adenine dinucleotide phosphate, reduced, ADP, adenosine triphosphate, PPI, inorganic pyrophosphate, CO2, carbon dioxide, SHMT1, serine hydroxymethyltransferase 1, SHMT2, serine hydroxymethyltransferase 2, MTHFD2L, methylene tetrahydrofolate dehydrogenase, NADP+ dependent, 2 like, MTHFD1L, methylene tetrahydrofolate dehydrogenase, NADP+ dependent, 1 like, MTHFD1, methylene tetrahydrofolate dehydrogenase, cyclohydrolase and formyl tetrahydrofolate synthetase 1, TDH, threonine dehydrogenase, GLY1, threonine aldolase, GCAT, glycine C-acetyltransferase, ALDH1L1, aldehyde dehydrogenase 1 family member L1, ALDH1L2, aldehyde dehydrogenase 1 family member L2, HAL, histidine. Ammonia lyase, UROC1, urocanid hydratase 1. AMD1, amidohydrolase domain containing 1, FTCD, formimidoyl transferase cyclodeaminase, MTHFR, methylene tetrahydrofolate reductase, MTR, 5-methyl tetrahydrofolate homocysteine methyltransferase, MAT1A, methionine adenosyltransferase 1A, MAT2A, methionine adenosyltransferase 2A, AHCY, adenosylhomocysteinase, CBS, cystathionine beta synthase, CTH, cystathionine gamma lyase, SDS, serine dehydratase, GCLC, glutamate cysteine ligase catalytic subunit, GSS, glutathione synthetase, PHGDH. Phosphoglycerate dehydrogenase, PSAT1, phosphoserine aminotransferase 1, PSPH, phosphoserine phosphatase, AGXT2, alanine glyoxylate and serine pyruvate aminotransferase 2, GRHPR, glyoxylate and hydroxypyruvate reductase, GLYCTK, glycerate kinase. Mild mitochondrial uncoupling is associated with increased longevity in nematodes 175 and fruit flies 176. Overexpression of uncoupling protein 1, UCP1, in skeletal muscle most greatly induced the expression of genes involved in glycine, serine, and asparagine synthesis, and 1-carbon metabolism 177, while the expression of pro-longevity cytoplasmic phosphenol pyruvate carboxykinase, PEPCKC, 178 was also induced. Overexpression of glycine N-methyltransferase, GNMT, which converts glycine and S-adenosyl methionine to sarcosine and S-adenosyl-homocysteine extended lifespan in Drosophila, and knockdown of GNMT prevented DR-mediated longevity 179. In mice, plasma sarcosine levels declined with age and increased with anti-aging CR 180. Metabolomics studies of aging using C. elegans have yielded fairly consistent data showing decreased levels of the vast majority of amino acids with aging 181,182. One group found glycine and aspartate levels increased in C. elegans with aging, while the levels of the other amino acids peaked in the late larval stages or first few days of adulthood and declined thereafter 183. Another group found glycine and alanine as the only amino acids to increase in abundance with aging 184, and a third group found glycine and glutamine to be the only amino acids that increased in abundance with aging, see Table 2, 184. DR resulting from the reduced pharyngeal pumping in E2 mutants prevented these increases in glycine and glutamine as well as prevented the decreased levels of arginine, valine, phenylalanine, and isoleucine, but not the decreased levels of the ketogenic amino acids lysine and leucine that occurred with aging 185. Glycine and serine supplementation have been shown to extend lifespan in C. elegans through stimulating 1-carbon metabolism including the methionine cycle 186. Glycine supplementation only extended lifespan at concentrations of 1 mm and below, Table 1, 3186. Knockdown of MEL32, the C. elegans homologue of serine hydroxymethyltransferase 1 and 2, see Fig 1, increased glycine levels and extended lifespan by preventing glycine conversion to serine and instead funneled glycine to the glycine cleavage system for 1-carbon metabolism including the methionine cycle. Consistent with this, knockdown of one of the 4 glycine cleavage system subunits, GCST1, increased glycine levels and decreased lifespan 186. 2.9. Histidine. Histidine is an essential amino acid that acts as an anti-glycating agent 187, free radical scavenger 188, and metal chelator 189,190. The dipeptide carnosine contains histidine and beta-alanine, is found at high levels in muscle and brain, and has similar physiological properties as histidine. But carnosine was shown to be less toxic in cell culture studies, so is a more promising therapeutic 187. Much data in model systems supports the potential use of carnosine for the treatment of aging-related disorders 191. But we will focus on histidine as the anti-aging effects of carnosine have been previously reviewed 192. Histidine supplementation increased insulin secretion and glycemic control, increased glutathione peroxidase activity, and decreased pro-inflammatory cytokine levels in diabetic mice 193. Histidine supplementation increased GSH levels and enhanced catalase activity to decrease liver injury induced by chronic alcohol consumption. 194. Individual administration of histidine, cysteine, or glycine, but not alanine, inhibited NF-kappa B activation in cultured coronary endothelial cells. 195. Fig. 1 shows that catabolism of histidine, cysteine, and glycine influence 1-carbon and redox metabolism. Histidine can scavenge singlet oxygen and hydroxyl radicals. 196. Low plasma histidine levels have been associated with inflammation, oxidative stress, and mortality in chronic kidney disease patients. 197. A metabolomics study of human plasma found histidine levels to decline with aging. 198. A metabolomics study of 647 human subjects linked high histidine levels, and 2 other amino acids, with longevity as defined by attaining at least 80 years. 199. In studies using Drosophila histidine supplementation showed no effect on the mean lifespan, while an equivalent dose of carnosine extended the lifespan of male flies. 200. However, histidine administration increased the lifespan of C. elegans at the 2 higher concentrations tested, but not at a lower 1 mm dose, Table 1, 3. The lifespan extension observed at the higher doses may have been due to its activation of the pro-longevity hypoxia-inducible factor 1, HIF1, and SKN1/NRF2 stress response pathways. 2.10. Isoleucine. Isoleucine, an essential amino acid that is both glucogenic and ketogenic, is one of 3 branched-chain amino acids, BCAAs, with leucine and valine being the other 2. 201. A metabolomics study showed that isoleucine and leucine levels decreased in the blood of aged human subjects. 202. Mice undergoing fasting, which is associated with anti-aging and anti-cancer properties. 203, showed increased plasma levels of all 3 BCAAs. 204. In a metabolomics study of young and aged rat brains, isoleucine levels increased, while tyrosine levels declined with aging. 205. Much conflicting data has been obtained regarding the role of BCAAs on metabolic health, insulin resistance, and glucose homeostasis. 206. In one study using rats, oral isoleucine was shown to decrease plasma glucose levels and stimulate glucose uptake into C2C12 myotubes in a phosphatidylainositol 3 kinase, Pi3K, dependent manner, while equivalent doses of leucine or valine did not. 207. Consistent with this, high blood levels of isoleucine have been associated with hypoglycemia in rats. 208. Altered isoleucine/leucine, indistinguishable by mass spectrometry, levels were found in aged Drosophila 82. Increased isoleucine levels in the culture media extended chronological longevity in yeast. 209 and just slightly increased lifespan in C. elegans. 3,23. In C. elegans, DAF16, a homologue of mammalian FOXO proteins, is a pro-longevity transcriptional regulator activated when insulin signaling is blocked. Metabolomics analysis of worms found that many amino acids, including isoleucine, glutamine, and valine were increased in long-lived DAF2 insulin receptor mutant worms, while glutamate levels declined. 210. Isoleucine and valine levels returned to wild-type levels in DAF2, DAF16 double mutants as did lifespan, suggesting a tight connection between these amino acids and longevity. However, isoleucine supplementation to C. elegans only increased lifespan at the highest 10 mm dose of 3 doses tested, and this effect was small. Table 1, 3. 2.11. Leucine. Like isoleucine, leucine is an essential BCAA. Leucine is one of 2 exclusively ketogenic amino acids and so is metabolized into acetyl-CoA and potentially into ketone bodies if blood glucose levels are low. A diet high in BCAAs has been shown to be associated with increased mean lifespan of male mice. 210. However, a diet low in BCAAs 211,212, or only low in leucine 213, increased glucose homeostasis and enhanced metabolic health in mice. High levels of the alpha-keto acid of leucine, alpha-ketoazocaproic acid, has been shown to cause mitochondrial dysfunction in neurons 214. A review of 34 reports in the literature has identified leucine supplementation to mice in a narrow range of 90 to 140 mg per day consistently protected metabolic health against diet-induced obesity, while BCAA administration had no effect 215. However, BCAA administration was protective in models of type 1 diabetes 215. BCAA levels increased with aging in mouse skeletal muscle 216. Leucine levels were found to decrease by 14% in the urine from aged rats and increase by 6% with exercise 217. Leucine strongly activates TOR kinase 218219 by binding cysteine 1 and cysteine 2 220, 221, 222 and by binding leucildernus synthetase 223224. Since inhibition of TOR kinase extends lifespan across eukaryotic species, one may expect leucine supplementation to decrease lifespan due to the downstream effects of TOR kinase activation including stimulation of protein synthesis and inhibition of autophagy 225. However, strikingly, to the best of our knowledge leucine supplementation has yet to result in decreased lifespan in any animal model yet tested, although a mouse diet high in BCAAs, and compensatorily lower in other amino acids, has been shown to decrease lifespan by decreasing the ratio of threonine and tryptophan to BCAAs, which led to decreased serotonin synthesis, hyperphagia, and obesity 226. Leucine is a major amino acid regulating the rate of protein synthesis by the ribosome in many tissues such as skeletal muscle 227228. Activation of TOR kinase increased levels of the YY1-PGC1 alpha complex in skeletal muscle to stimulate mitochondrial oxidative function to match energy supply with energy demand 229. Administration of leucine has been shown to decrease muscle degeneration in vitro 230 and may increase overall muscle anabolism in young to middle-aged healthy humans 231,232. Leucine supplementation has been shown to promote fatty acid oxidation, stimulate mitochondrial biogenesis, increase NAD+ levels, and upregulate SIRT1 and AMP kinase, AMPK, activity in skeletal myotube cells 233. Although leucine is commonly consumed by young or middle-aged athletes to facilitate an increase in muscle mass, there is little evidence to support the long-term use of leucine supplementation without exercise in the elderly for the amelioration of sarcopenia 234,235. A study in Drosophila showed that lowering BCAAs in the diet extended lifespan, but not more than a diet lowering 3 other essential amino acids threonine, histidine, and lysine 236. So at least in fruit flies, the BCAAs do not suppress longevity more than other essential amino acids. Restriction of BCAAs did not further extend lifespan of an already low amino acid diet, suggesting that BCAA restriction and general protein/amino acid restriction activate the same molecular mechanisms. One group showed that individual supplementation with leucine could extend chronological longevity in yeast 209 and that leucine availability was important for maximal lifespan extension during CR 237. However, another group under different conditions showed that blocking leucine synthesis in yeast stimulated chronological longevity 238. Leucine supplementation led to a prominent increase in lifespan in C. elegans that was slightly greater than valine and much greater than isoleucine, Table 1, 3,23. The extended longevity could be mimicked by knocking down the BGAT1-BCAA aminotransferase 23. Surprisingly, knockdown of BGAT1 in neurons also extended lifespan that was reduced by concurrent neuronal knockdown of TOR kinase, LET 363, 23. The lifespan extension resulting from BGAT1 knockdown or leucine supplementation could also be mitigated by ablation of ACI neurons, perform some functions of the mammalian hypothalamus. This data suggests that neuronal-TOR has a pro-longevity function. In contrast to these results, long-lived global mTORC1 pathway-deficient worms lost their enhanced longevity and became normal-lived when mTORC1 activity was restored specifically in neurons, but not when mTORC1 activity was specifically restored in the intestine 13. This data indicates that neuronal TOR has a pro-aging function. Perhaps decreased function of both mTORC1 and mTORC2 by TOR kinase/LET363 knockdown in the first example above explains the divergent results. It has been shown that specific mTORC1 deficiency compared to concurrent mTORC1 and mTORC2 deficiency led to the activation of different stress response pathways 239. The inability to restore normal lifespan by restoring mTORC1 activity in the intestine at first glance appears to be inconsistent with other data where specific inhibition of mTORC1 in the intestine extended lifespan 239. But it is possible that specific knockdown of mTORC1 activity in the intestine generates a signal inactivating mTORC1 activity in neurons that leads to lifespan extension, which is consistent with the other findings. 2.12. Lysine. Lysine is an essential ketogenic amino acid. Therefore both leucine and lysine can be supplemented while a subject is on the anti-aging ketogenic diet 240,241 without stimulating gluconeogenesis to raise glucose levels. A metabolomics study of 647 human subjects linked high lysine levels, and 2 other amino acids, with longevity as defined by attaining at least 80 years 199. In humans, lysine supplementation was shown to mimic protein supplementation to increase plasma insulin and glucagon levels, and when lysine was administered with glucose it decreased blood glucose levels 242. Lysine supplementation also decreased blood glucose levels and prevented cataract formation when administered to diabetic rats. An equivalent amount of a mixed amino acid supplement was also beneficial for both of these disease phenotypes, but not to the extent of lysine 243. Lysine supplementation induced an angiogenic response and increased the rate of wound healing 244. In yeast, deletion of the lysine biosynthesis gene Lys12 increased chronological lifespan 93. Lysine was the only amino acid to increase in abundance in replicatively aged yeast, while the abundance of roughly half of the amino acids declined 95. In C. elegans, lysine supplementation yielded small increases in lifespan at all 3 doses tested, Table 1, 3. 2.13. Methionine. Reducing dietary levels of methionine, an essential amino, in rodents was shown to partially mimic the effects of CR and induce lifespan extension 245. This topic has been extensively reviewed 246, 247, 248, 249, 250, 251. Many of the protective effects of DR are blocked by adding back methionine to the diet 252. Some of the protective effects of CR and methionine restriction are due to increased transsulfuration pathway flux to increase the expression of the cystathionine gamma lyase, CTH, gene and the production of hydrogen sulfide, H2S, 252,253. Not only can CTH catabolize cystathionine to cysteine, but it can catabolize cysteine to H2S 253. Cysteine supplementation can inhibit some of the beneficial effects of methionine restriction 254, perhaps through restoring glutathione levels that are decreased during methionine restriction 255. The decreased glutathione levels led to the activation of protein kinase R, like endoplasmic reticulum kinase, PERK, and the integrated stress response, ISR, including transcription of the ATF4 transcriptional regulator 256. Increased hepatic ATF4 levels stimulated release of fibroblast growth factor 21, FGF21, leading to increased expression of uncoupling protein 1, UCP1, in white adipose tissue, WAT, and a browning of Wa 256, which results in increased energy expenditure. FGF21 has been shown to be transported into the brain and signal to alter metabolism during a low-protein diet 257. It also likely plays a similar role during methionine restriction. The NRF2 transcriptional regulator was also activated by methionine restriction, likely due to the decreased glutathione levels. NRF2 increases transcription from antioxidant response elements, AREs, in gene promoters. Dietary methionine restriction inhibits mTORC1 activity to activate autophagy by lowering tissue levels of S-adenosylmethionine, which normally binds the CIMTR protein to stimulate mTORC1 activity 258. In yeast, increased methionine and S-adenosylmethionine levels also inhibit autophagy by stimulating the S-adenosylmethionine-dependent methylation of the catalytic subunit of the pp2A phosphatase by the ppm1p methyltransferase, leading to the dephosphorylation of the NPR2P regulator of autophagy 259. Therefore, decreased S-adenosylmethionine levels, inhibition of mTORC1, and activation of autophagy likely explain the lifespan extension observed following knockdown of the S-adenosylmethionine synthetase 1, SAMS1, gene in C. elegans 260, even though stimulation of the methionine cycle is also associated with longevity under other conditions. 186. Methionine restriction can also occur during a vegan diet 261,262 or a strict ketogenic diet 263 due to the low protein intake prescribed to these diets, and the addition of methionine to a ketogenic diet resulted in reversal of weight loss in mice 263. A vegan diet is also typically lower in lysine content 264. A diet deficient in methionine is associated with many positive effects such as enhanced fatty acid oxidation, increased energy expenditure, decreased ROS production, reduced oxidative damage 265, and inhibition of tumorigenesis 266. Most notably, methionine restriction extended lifespan in mice by nearly 7% 267, rats by 44% 268, and Drosophila by 36% 269. Similar to CR, methionine restriction produced a lifelong reduction in fatty body mass 245. A diet that restricts methionine by 83% in humans, similar to the extent that produces the positive longevity effect in rodents, has been established. 266. Glycine supplementation to rats mimicked the longevity effects of methionine restriction, though stimulating the clearance of hepatic methionine. 270. Methionine restriction had greater benefits to the metabolic health of mice than equivalent leucine restriction. 271. When fruit flies were placed on a protein-deficient diet that extended lifespan, the addition of methionine could restore fecundity without decreasing lifespan, suggesting that it is possible to design a modified amino acid diet that offers the positive effects of DR without the negative effects. 4. Methionine restriction also extended lifespan in Drosophila 4, but only on a low amino acid diet. 272. In one study in yeast, chronological lifespan was extended by methionine restriction, but not by lysine, isoleucine, threonine, or valine restriction. 273, and this increased longevity required autophagy-dependent vacuolar acidification. 274. Methionine restriction extended the chronological lifespan of yeast even when H2S production was inhibited. 275. Methionine supplementation increased mitochondrial oxidative metabolism in yeast. 276, and also increased oxidative stress resistance by increasing pentose phosphate pathway flux and cellular reduced nicotinamide adenine dinucleotide phosphate, NADPH, levels. 277. Methionine supplementation to fasted mice and catabolism of the methionine through the little-studied methionine transamination pathway decreased blood sugar levels by inhibiting hepatic gluconeogenesis through GCN5-acetyltransferase and PGC1 alpha-dependent mechanisms. 278. Methionine supplementation was shown to extend the lifespan of C. Elegans at the two highest doses administered, but not at the lowest dose, Table 1, 3. 2.14. Phenylalanine. Phenylalanine is an essential amino acid that is both glucogenic and ketogenic and is converted to tyrosine by phenylalanine hydroxylase. Phenylalanine and glutamine levels were found to be higher in the plasma of centenarians compared to other elderly individuals. 279. Using a longitudinal approach, phenylalanine/tyrosine metabolism, tryptophan metabolism, and methionine metabolism were shown to be altered with aging in the plasma of marmosets, a primate aging model. 280,281. Specifically, phenylalanine levels decreased with age. 281. Phenylalanine binds hydroxyl radicals and prevented hydroxyl radical-mediated inhibition of acetylcholinesterase activity in brain homogenates. 282. Elevated serum phenylalanine has been linked to telomere loss in men. 283, inflammatory disease. 284, and type 2 diabetes. 33. Dietary protein restriction in rats increased lifespan and decreased phenylalanine levels in liver. 285. Increased phenylalanine levels were found in aged Drosophila. 82. Increased dietary protein as well as individual supplementation with phenylalanine, methionine, serine, or threonine greatly decreased the lifespan of the Argentine ant, while individual supplementation with most other amino acids slightly decreased lifespan and supplementation with glutamate, tyrosine, or tryptophan did not affect lifespan. 286. Phenylalanine was one of two amino acids that did not extend the lifespan of C. elegans when added in the 1 to 10 mm range and was the only amino acid that decreased lifespan at the 5 mm dose, Table 1, 3. Phenylalanine can be oxidized to the toxic metabolite metatyrosine, M-tyrosine, which decreases C. elegans lifespan. So during oxidative stress, C. elegans upregulates the expression of tyrosine aminotransferase, TATHUN1, to deaminate excess tyrosine, which likely decreases product inhibition of phenylalanine hydroxylase to stimulate the conversion of phenylalanine to tyrosine and decrease phenylalanine and M-tyrosine levels. 287. 2.15. Proline. Proline is a non-essential amino acid that is synthesized from glutamate in a two-step NADP-requiring pathway. A metabolomics study of aging mice found proline, alanine, serine, tyrosine, and methionine to decrease in plasma with aging. 53. Increased plasma proline concentrations have also been associated with sarcopenia in the elderly. 288. Dietary proline supplementation had an immunostimulatory effect to protect mice from Pasteurella multocida infection. 289. The topical application of proline to cutaneous wounds led to an acceleration of healing in rats. 290. Proline acts as a metal chelator, supports osmotic balance, and prevents oxidative damage. 291. Proline supplementation mitigated the early stage of liver injury in rats by decreasing oxidative stress. It also improved redox status to improve nitric oxide availability and prevent a pathological increase in blood pressure. 292. Proline was the second most potent amino acid at extending lifespan in C. elegans, Table 1, and the most potent for increasing thermotolerance. 3, suggesting that it stimulates pathways mediating protostasis. It has been shown to extend the lifespan by transiently increasing ROS levels, which then stimulate stress response pathways that induce antioxidant gene expression. 293. Proline is hydroxylated to form hydroxyproline, a prominent amino acid in collagen, a major protein of the C. elegans cuticle. 294. Collagen remodeling was shown to be required for C. elegans lifespan extension due to DAF2 insulin receptor mutation. 295. 2.16. Serine. Serine is a non-essential amino acid that is synthesized in a three-step pathway from the glycolytic intermediate 3-phosphoglycerate, Fig. 1. Serine supplementation showed many protective effects on the phenotypes induced by a high-fat diet in mice, including increasing glucose tolerance and insulin sensitivity, decreasing hepatic lipid levels, and preserving reduced glutathione, GSH, levels by preventing the hypermethylation of promoters of GSH synthesis genes 296. Serine supplementation also activated AMPK in palmitic acid-treated primary hepatocytes to decrease ROS production 296, 297, 298, 299. Although short-term serine supplementation did not reduce weight gain on a high-fat diet 296, long-term serine supplementation to a normal chow diet was shown to decrease food consumption and weight gain 300. Serine supplementation increased SIRT1 protein deacetylase expression and decreased ROS levels in the hypothalamus, and decreased levels of phosphorylated NF-kappa B to decrease the levels of pro-inflammatory cytokines 300. Serine has been shown to be neuroprotective 300 and 1302 in part through upregulation of the expression of ER protein disulfide isomerase to increase protostasis 300 and 3304. With aging, microglia synthesize and release increased amounts of D-serine, synthesized from L-serine. D-serine is a coagonist for NMDA receptors 305 and therefore this increased extracellular D-serine may contribute to the excitotoxicity induced by neuronal NMDA receptor activation in aged animals 306. A phase I clinical trial has been conducted testing the effects of serine supplementation on amyotrophic lateral sclerosis, ALS, patients 307,308. A 15g dose of serine twice a day was well tolerated with only mild intestinal discomfort in a few subjects. It has been postulated that the long life of Ojibwe village Okinawans may be due in part to the high content of serine in their diet 309. Serine metabolism can increase cellular antioxidant function through several different mechanisms. In studies with human endothelial cells, serine supplementation increased antioxidant gene expression through upregulation of NRF2 and its downstream effector heme oxygenase 1 310. Serine is a direct precursor for the synthesis of glycine and cysteine, which are limiting for the synthesis of GSH. See Fig. 1. Once GSH is oxidized, the GSSG is reduced by NADP and recycled for use. 1 carbon units derived from serine can also be added to homocysteine for methionine synthesis. Serine is transported into the mitochondrial matrix by the SFXN1 transporter 311. Catabolism in mitochondria, initiated by the enzyme SHMT2, increased mitochondrial NADP levels to increase antioxidant function 312. Data has emerged that mitochondrial serine can also be catabolized in a 3-step pathway to 2-phosphoglycerate, Fig. 1, which can then be exported to the cytoplasm for gluconeogenic or glycolytic metabolism 313,314, while synthesis of serine from glycolytic 3-phosphoglycerate occurs in the cytoplasm 315,316. Serine is also an essential building block of lipids such as phosphatidylserine 317, sphingolipids, and ceramides. Serine deficiency results in mitochondrial fragmentation and dysfunction 318. Anti-aging CR was shown to increase serine dehydratase levels in the liver of aged mice to increase the catabolism of serine to pyruvate to fuel gluconeogenesis 319. Subjects with fibromyalgia showed lower plasma levels of serine and histidine 320. In yeast inhibition of serine synthesis from 3-phosphoglycerate, see Fig. 1, increased chronological lifespan by stimulating glyoxylate shunt activity and Trehalose synthesis 321. In other studies with yeast, serine was further shown to promote aging by activating a phosphorylation signaling cascade using the PKH1/2, SCH9, homologue of mammalian S6 kinase, and RIM15 protein kinases 322. In contrast to the results with yeast, in C. elegans serine supplementation increased lifespan to the greatest extent of any amino acid, Table 1, 3. Serine supplementation activated the HIF1, DAF16, and SKN1 stress response pathways, and lifespan extension was dependent upon the function of AAK2/AMPK and SIR2 and SIR2.13. 2.17. Threonine. Threonine is an essential amino acid that is both glucogenic and ketogenic. Threonine supplementation in humans can greatly increase its levels in plasma. 323. Threonine is a precursor for glycine synthesis in most mammals, and threonine supplementation has been shown to increase plasma glycine levels in pigs. 324 and increase glycine neurotransmitter levels in the brain of rats. 325. An excess or deficiency in dietary threonine decreased protein synthesis rates in pigs. 324. Threonine levels were shown to decline in the plasma of aged rats. 205. A metabolomics study of 647 human subjects linked high threonine levels, and 2 other amino acids, with longevity as defined by attaining at least 80 years. 199. In human 326 and mouse embryonic stem cells, threonine levels can regulate the cell cycle G1/S phase transition and proliferation. 327,328. Threonine deficiency in mammals has been associated with depression and neurological dysfunction. 329. Threonine supplementation to chickens led to an improved immune response. 330 and protection from LPS-induced inflammation and intestinal barrier damage. 331. Threonine supplementation to pigs led to increased protection against a viral challenge. 332. Supplementation with threonine has been shown to extend chronological longevity in yeast. 209. However, a different group using different conditions found threonine restriction, as well as valine restriction, to increase yeast chronological lifespan. 322,333,334 by inhibiting TOR kinase. 322. Threonine supplementation only extended C. elegans lifespan slightly at the highest, 10 mm, dose of 3 doses applied, Table 1. Threonine can be converted to glycine in 1 step by threonine aldolase and in 2 steps by the threonine dehydrogenase and GCAT enzymes, Fig. 1. Therefore threonine could extend lifespan by a similar stimulation of the methionine cycle as shown for glycine or serine supplementation. 186. However, another mechanism could be involved. The 2-amino-3-ketobutyrate product of the threonine dehydrogenase reaction is unstable and spontaneously decarboxylates into aminoacetone, which is further broken down producing hydrogen peroxide and methylglyoxal. Knockdown of the C. elegans GCAT homolog T25B 9.1 extended lifespan through increasing 2-amino-3-ketobutyrate and the production of hydrogen peroxide and methylglyoxal, which then activated the SKN1 and HSF1 transcriptional regulators. So lifespan was extended by a hormetic effect. 335. Therefore, increased production of these reactive products could also contribute to the lifespan extension that occurs during threonine supplementation. 2.18. Tryptophan. Tryptophan is an essential amino acid that is both glucogenic and ketogenic. Dietary tryptophan restriction in rats delayed reproductive aging. 336 and extended lifespan. 337,338. However, the rats were switched to a normal diet later in life, so more experiments need to be done to confirm these lifespan findings. 339. Tryptophan is a precursor for the neurotransmitter serotonin and for the hormone melatonin that regulates circadian rhythms. Tryptophan is degraded by the kynurenine pathway to nicotinamide adenine dinucleotide, NAD+, and other end products, with several of the intermediates being toxic such as quinolinic acid, due to it being an agonist of the neuronal NMDA receptor. The intermediate kynurenic acid is neuroprotective because it is an antagonist of the NMDA receptor. Tryptophan levels were shown to decrease in human serum with aging. 198, while toxic tryptophan catabolites were shown to increase. 340. This increase in tryptophan catabolism with aging is likely due to increased levels of the enzyme indoleamine 2,3-dioxygenase, IDO, 340. IDO breaks down tryptophan to kynurenine in tissues outside the liver, while tryptophan 2,3-dioxygenase performs this task in the liver. IDO is induced by pro-inflammatory cytokines and superoxide 340, which increase with aging. Tryptophan plasma levels have been measured or tryptophan supplementation studies have been performed for many disorders as recently reviewed 341. In brief, studies have been performed focusing on tryptophan in the following aging-related disorders: cardiovascular disease 342, 343, 344, chronic kidney disease 345, diabetes 346, depression 347,348, inflammatory bowel disease 349, and multiple sclerosis 350. Tryptophan levels were most frequently lower in the disease state and supplemental tryptophan most often decreased disease phenotypes. There were two exceptions. Limiting tryptophan was beneficial for chronic kidney disease, and increased plasma tryptophan levels were found to predict future type 2 diabetes in a Chinese population 346. However, this association with type 2 diabetes has yet to be found in other ethnic groups. In fact, several tryptophan catabolites have been found to be increased in the plasma of subjects with type 2 diabetes 351, suggesting increased tryptophan catabolism. In rats, tryptophan supplementation was shown to lower blood glucose levels, and delay disease progression in hereditary diabetic rats 352. A decrease in plasma tryptophan levels with aging was associated with a loss of cognitive function in non-demented women 340. Supplemental tryptophan was also shown to improve the memory of aged rats 353, while tryptophan-enriched cereals were shown to improve the sleep/wake cycle of elderly humans 354. Altered tryptophan levels were found in aged Drosophila 82, and long-lived flies showed higher tryptophan levels 355. Decreasing tryptophan uptake by deleting the tryptophan permease TAT2P increased replicative lifespan in yeast 356. Blocking tryptophan catabolism extended lifespan and decreased aging-related proteotoxicity in C. elegans 22. Tryptophan supplementation increased the lifespan of C. elegans at two lower doses, but not at the highest 10 mm dose tested, Table 1, 3. Tryptophan was the only lifespan-extending amino acid tested out of 10 tested that extended the lifespan in DAF16 mutant worms. Also, tryptophan and cysteine were the only two lifespan-promoting amino acids out of 10 tested that did not activate a transcriptional reporter for SKN1/NRF2 activity, suggesting novel mechanisms of lifespan extension. 3. Tryptophan and a few of its catabolites were shown to activate the mitochondrial unfolded protein response, UPRMT, and the ER stress response pathways in C. elegans 3. The UPRMT is frequently, but not always, associated with nematode longevity 357,358. 2.19. Tyrosine. Tyrosine, a non-essential amino acid, is both glucogenic and ketogenic and is synthesized from phenylalanine by the phenylalanine hydroxylase enzyme. Tyrosine is the precursor for the neurotransmitters dopamine and norepinephrine. In a metabolomics study of human plasma, tyrosine and glutamine levels increased with age, while histidine, threonine, tryptophan, leucine, and serine decreased with age. 359. Supplementation with tyrosine in both hypertensive humans and rats led to a decrease in blood pressure. 360. In one study of human subjects, increased dietary tyrosine levels were linked with increased cognitive performance. 361. However, another study found that supplementation of moderate tyrosine levels had no effect, while high dietary tyrosine levels decreased cognitive performance in older adults. 362. Authors of a recent review suggest that tyrosine supplementation likely only increases cognition during times of stress when either dopamine or norepinephrine levels are depleted. 363. There is no consistent data that tyrosine supplementation can decrease the effects of stress on cognitive or physical performance. 364. Tyrosine supplementation has been shown to increase the vasoconstriction response to body cooling in older adults, so it may aid thermoregulatory function. 365. Tyrosine supplementation has been shown to enhance dopaminergic neurotransmission in Parkinson's disease patients. 366. However, high levels of tyrosine in the plasma increase the risk of type 2 diabetes to a similar extent as increased levels of each of the three BCAAs. 33. In C. elegans, tyrosine supplementation increased lifespan moderately at the lowest 1 mm dose, but only slightly at the two higher doses. Table 1, 3. 2.20. Valine. Valine is an essential glucogenic BCAA. A breakdown product of valine, 3-hydroxyisobutyrate, 3-HIB, was shown to drive vascular fatty acid transport and insulin resistance. 367. However, valine supplementation to rats reduced fatigue during swim exercise and partly prevented the drop in blood glucose and liver glycogen following exercise, while leucine or isoleucine supplementation did not. 368. Valine supplementation also protected against paraquat-induced toxicity in rats. 369. Valine supplementation, more than the supplementation of any other amino acid, increased ATP levels and protected against oxidative stress in M17 neuroblastoma cells overexpressing alpha-synuclein 100. This could be due to an increase in mitochondrial biogenesis induced by BCAAs 210, as isoleucine showed a similar, but smaller effect. 100. The mitochondrial carrier family member SLC25A44 has recently been identified as a mitochondrial BCAA transporter that plays an especially important role in brown adipose tissue metabolism. 370. Using yeast, one group showed that supplemental valine extended chronological longevity. 209. However, another group using different conditions found valine restriction to increase yeast chronological lifespan by inhibiting TOR kinase. 322,333,334. Replicative aging in yeast has been shown to lead to an increase in the expression of amino acid catabolism genes, a decrease in the expression of amino acid biosynthesis genes, and a decrease in most amino acid levels, especially all three BCAAs but also asparagine, glutamate, glycine, histidine, and methionine. See Table 2, 95. In C. elegans, valine supplementation increased lifespan moderately at the two lower 1 mm and 5 mm doses, but not at the highest dose. Table 1, 3. 3 Conclusions. In the last decade, major strides have been made in the understanding that amino acids are signaling molecules and can be used as a relatively non-toxic treatments to alter the rate of aging in experimental models. Important molecular details of how specific amino acids such as leucine and arginine activate TOR to decrease lifespan have been elucidated. But the molecular mechanisms through which increased levels of many of the amino acids provide health benefits remain largely unknown. The ability of glycine to extend longevity in rodents is an important breakthrough, as amino acid restricted diets, such as methionine restricted diets, are expensive and can be relatively unpalatable, resulting in difficulties in subject retention in clinical trials. Another important goal should be the determination of the effects of serine and threonine on the lifespan of mice, since these amino acids can be metabolized to glycine but also have their own unique properties. In support of these unique properties, serine or glycine administration to rats altered brain amino acid levels in different ways. 371. Women have been shown to have higher plasma levels of serine and glycine than men, while 13 of the other 18 amino acids were lower. 359. Whether these amino acid level differences contribute to the extended longevity of women compared to men remains to be determined. Serine supplementation in humans has been shown to lower plasma homocysteine levels, a marker of metabolic and cardiovascular disease 372. This may occur through stimulation of cystathionine beta synthase, CBS, activity, see Fig 1. Serine metabolism in mitochondria leads to NADP production that protects mitochondria from ROS-mediated damage, but at the expense of NADP oxidation in the cytoplasm 373. However, some of these lost cytoplasmic reducing equivalents can be restored if increased serine levels stimulate CBS activity of the transsulfuration pathway to increase cysteine levels, which decreases the plasma membrane import of oxidized cysteine and its subsequent reduction to cysteine, which is indirectly coupled to cytoplasmic NADP oxidation 374. The human threonine dehydrogenase 375 and threonine aldolase 376 genes have mutated so functional proteins are not expressed, while the expressed proteins are functional in rodents for the conversion of threonine to glycine, Fig 1, 373. So threonine supplementation to humans would not likely yield similar effects to those of glycine. Several amino acids from the diet are catabolized to a large extent by first-pass splanchnic 377 and hepatic 378 metabolism, and amino acid supplementation strategies may fail to raise amino acid levels to a large extent in peripheral tissues due to this issue. Since the hypothalamus is a major regulator of the aging process, amino acids may need to cross the BBB to exert their anti-aging effects, and in doing so they must compete with other amino acids for transport 158. So it may be difficult to raise the level of many amino acids in the brain. To address these issues, studies could be performed with membrane-permeable forms of amino acids such as N-acetylated forms and ethyl ester forms that may more readily permeate throughout the body. Another challenge that needs to be addressed is determining the correct amount of an amino acid to supplement to obtain a therapeutic effect, as a hormetic dose response is often observed. 3. The presence of transaminases and alpha-keto acids in the blood and many tissues including the liver lead to the relatively quick transamination of one amino acid and synthesis of another. Many of the common transaminases such as aspartate transaminase and alanine transaminase use alpha-ketoglutarate as an acceptor of an amino group to form glutamate. Therefore, the effects of supplementation with certain amino acids may be a result of increased glutamate or decreased alpha-ketoglutarate levels instead of from a direct effect of the supplemented amino acid. In this regard, alpha-ketoglutarate has anti-aging properties 140 even at low doses 3, and so supplementation with high levels of certain amino acids may shorten lifespan through alpha-ketoglutarate depletion. Amino acid transamination likely explains why 18 of the 20 amino acids extended lifespan in C. elegans and why many of the amino acids demonstrated a similar hormetic dose response. 3. At high doses of supplementation, a common mechanism of toxicity may be occurring even though a different amino acid was supplemented. Heavy isotope amino acid tracer studies will be useful to follow the metabolism of the supplemented amino acids to determine if common downstream pro-longevity and pro-aging catabolites can be identified as causative longevity modulators. Therefore, much work remains to harness the great potential of altering amino acid levels for the treatment of aging and aging-related diseases.